Hi everyone, in this slide, it's normally talk about the sample preparations in X-ray diffractions, XRD. So the sample preparations in XRD involve 5 parts. First, drying and powdering solid sample. Second, mounting smear slide for small sample. Third, removing carbonated from high carbonated sample. Fourth, separating the clear fractions from larger grain size particle. And lastly, expanding swirling clear with ethylene glycol treatment. So the first part is the grinding solid sample. So solid samples are prepared for X-ray diffractions by grinding, which can be accomplished by several different methods depending on the sample matrix, the size of the sample, and quantity of prepared material needed. First, we need to freeze dry sample for at least 12 hours before grinding. Grind solid sample to a fine tar-like powder by using one of the methods, which is spec shelter box. Next, we transfer the powder sample to an appropriate labeled glass bottle, followed by select the steel or plastic sample holder unless the sample material is very small. Lastly, place enough powder from the labeled sample bottle to fill an empty sample holder. Gently press the powder flush with the sample holder using a glass slide. Next, we move to part 2, which is sample slurry or small slide marked for small sample amount. So first, we grind the sample to a tar-like powder, which is less than 0.062 mm. Next, we place a small amount of sample in the center of the quart dish. 2 squash dish will adequately fill the sample holder, while the upper squash dish will be used for the slurry. Next, we add 2 or 3 drops of acetone or distilled water to the sample. Please be noticed that acetone dries faster than water. Next, we spread the sample to a thin layer with a glass rod. This is because rolling it over the sample works well. And lastly, we place the sample in the desiccator to dry. After that, we move to the part C. We need to remove the carbonated before clear separations. It may be necessary to dissolve the carbonated in some sediment before the clear mineral can be identified. So the goal is to remove as much carbonated as possible to isolate the material contained within the carbonated for analysis. So in these cases, there are 2 types of treatment. The first one is the hydrochloric acid treatment, and the second one is the acetic acid treatment. But in these cases, we are using the acetic acid treatment instead of hydrochloric acid treatment. This is because hydrochloric acid treatment is a treatment with strong acid to remove carbonate, which can attack the structure of the clear mineral, particularly trioctahedral mineral, compared with the acetic acid treatment, which is slightly more involved method. So first, we are placed around 2 cm³ of undried sample into a centrifuge tube with 25 ml of acetic acid, which is 10% of the solutions. Mix well and let sit until the reaction ceases. Shake well again to ensure the reactions has stopped. For example, no more bubbles observed. Spin sample in the centrifuge for 50 minutes at 1500 resolutions per minute. Decant the acetic acid solutions and dispose of the acid solution properly. Next, we add 25 ml of DI to the centrifuge tube and centrifuge again for 50 minutes at 1500 resolutions per minute. Decant the clear water. Repeat the wash cycle from step 4 to 6 with DI. Place wash sample in a large beaker with some distilled water and a little 1% coagulant. Suspend the clear material by placing the beaker in a sonic decelerator for around 1 minute. Please be noticed that do not let the sample heat up. Next, we transfer the sample to a clean centrifuge tube and spin for 5 minutes at 1000 revolutions per minute to remove the more than 2 micrometer size fractions from suspension. Remove the less than 2 micrometer size fractions by collecting the top 1 centimeter cube of solutions with an eyedropper. Lastly, make an oriented clay mold by placing 2 or 3 drops, which is enough to cover the quartz dish of clay suspension, onto a glass slide and let dry in a desiccator. Next, we move to the fourth part, which is separating the clay. So there are various methods for separating clay from coarse material. Those listed below are methods used on board. For example, the quick and dirty clay separation method. First, in centrifuge tubes, mix a small amount of bulk sample, which is around 5 ml, with 1% of coagulant solutions. Notice that we need to use an ultrasonic bath or dismembrator if necessary. Centrifuge the coagulant solutions or sample mix at 1000 revolutions per minute for 5 minutes to remove the more than 2 micrometer particle size fractions. Decant the coagulant solutions containing suspended clay into a new centrifuge tube and spin it at 1500 rpm for 15 minutes to remove the remaining less than 2 micrometer clay size fractions. Next, we decant the coagulant solutions and wash the clay residue with distilled water. Spin down again at 1500 revolutions per minute for 15 minutes. Lastly, make an oriented clay mold by placing 2 to 3 drops or enough to cover the quartz dish of solution onto the quartz dish and let dry in the desiccator. Last but not least, the last part for the sample preparation in XRD is the treating with ethylene glycol. So ethylene glycol can be used to expand swirling clay, some mixed layer clay, and vermiculite as an aid to mineral identifications. So there are 2 ethylene glycol treatments methods, which is vapor treatments and quick treatments. So in these preparations, we are using the vapor treatments instead of the quick treatment. This is because the vapor treatment is less disturbance of the sample and less amorphous scattering of X-ray by excess liquid than in the quick method. So first, we find the glyconator container stored in the ICP pre-sinks cube board. Pour the ethylene glycol to a depth of around 1 centimeter cube in the bottom of the container, and then we place the sample to be glyconated onto the shelf. Place the container, which is the glyconator, in oven at 60 degrees Celsius to 70 degrees Celsius overnight. And lastly, keep the sample in glyconator until ready to analyze. So these are the steps involving in sample preparations in XRD. Hope you guys will get some knowledge about it. Thank you.